Okay, now that we're done with our probabilities laws, it's time for us to look at a question that allows us to analyze data using chi-square. So number 12 says Dr. William Cresco, a researcher at the University of Oregon, conducted several crosses between marine stickleback fish and freshwater stickleback fish. All marine stickleback fish have spines that protrude from the pelvis, which presumably serve as protection from larger predatory fish. Many freshwater stickleback populations lack a pelvic spine. Dr. Cresco wanted to find out whether the presence or absence of pelvic spines behaves like a Mendelian trait, meaning that it is likely to be controlled mainly by a single gene. In one cross, marine sticklebacks with spines were crossed with sticklebacks from Bear Pond Lake, which don't have pelvic spines. All the progeny fish from this cross, the F1 generation, had pelvic spines. Dr. Cresco then took the F1 offspring and conducted several crosses between them to produce the F2 generation. The results of the F2 crosses are shown in the table to the right. So I underlined some important information from this text. So in red, I've identified the purpose for this investigation. It was to determine whether or not the presence or absence of pelvic spines behaves like a Mendelian trait, meaning that it is controlled mainly by a single gene. Also, when we hear that word Mendelian trait, we know that that is where one allele is completely dominant over another allele. So that is the purpose of the investigation and the hypothesis that we will be testing. In blue, what I've underlined there is that all the progeny fish from this cross, the F1 generation, had pelvic spines. When we hear that word F1, we know that we should be thinking of a hybrid or a heterozygote. So all the progeny from that cross, the F1 generation, having spines shows us that pelvic spines are dominant because all of the heterozygous fish have pelvic spines. So then finally, the data that we're going to be analyzing is the results of an F2 cross. So if you look where it says table 14, it says F2 generation, a cross of F1 generation individuals. So this means that we are crossing two F1s, and all of these offspring are from the F2 generation. Now we're ready to start our chi-square analysis. What I have at the very top is right from our AP Bio equation sheet is the chi-square equation, and then we also have the chi-square table, which we're going to need to use to analyze our results. On your guided notes, you do not have a table. You're not required to have a table. Sometimes AP provides a table, many times they do not. I think the table helps organize our data. If you want to use a table, please go ahead and copy this table onto your notes. Okay, so our first important data that we need to recognize is what are our observed values. Our observed values are always provided in the question. We need to be told what the observed values are, and this is the data that the scientists collect. Notice I put a red box around the data that we're going to look at. Whenever we do statistical analyses, we're not going to look at one cross or one trial. We're going to look at our total data from all of the trials that were conducted. So these scientists conducted 12 different trials. So we're going to look at those total amounts. So we're looking at the F2 fish with spines, that totaled 320 fish with spines, and then the F2 fish without spines, and that totaled 88. So those are going to be our observed values. As you can see here, I've filled that into our chi-square equation. 320 go with the spines, and 88 go without spines. That's always the easiest thing to do in chi-square analysis, is to determine the observed. What is much harder is to determine the expected values. So to determine those expecteds, I'm going to ask you to come back and look again at what I've underlined in red. Our purpose here is to see if this trait behaves like a Mendelian trait. Mendelian traits mean that there's one gene and one allele is completely dominant over the other. So because the F1 phenotype had spines, I made a key and my dominant allele is going to be spines. That's the phenotype it's going to represent. Also, the recessive allele is then going to be spineless. And remember, that's determined by the phenotype of our F1. We know our F1 is heterozygous. We now need to determine what are our expected values for our F2 generation. So the F2 generation is a cross of two individuals from the F1 generation. So I'm crossing two heterozygotes. Notice after I do my Punnett square, crossing two F1s, I have 3/4 of those individuals who are going to have spines, the homozygous dominant and the two heterozygotes, and then only 1/4 will be spineless. We are going to use those percentages or those fractions to determine our expected values. So one thing that I want to show you here, the total number of fish that we had was 408. I got that 408 because we had 320 fish with spines, and I added that to the 88 fish without spines. So we have a total of 408 fish. It is very important that you always take your fractions or your percentages and you multiply that by your total population, not just the fish with spines. We predict that 3/4 of the entire population will have spines. So we have to take 3/4 and multiply it by 408. When we do that, we have an expected value of 306. And then without spines, we believe that that probability will be 1/4. So we multiply 1/4 times 408, and that probability, we would expect 102 fish to be without spines. Right now, I would like you to fill out the next column, your observed minus expected values for spines and without spines. Okay, if you did that correctly, you should have a 14 for spines for your observed minus expected, and you're going to have a negative 14 for without spines. Don't worry about that negative. Remember that we're going to take those values and we're going to square them, and then also divide by our expected values in the next column. So I'd like you to do that now. So if you did that correctly, when you square 14, you get 196. Notice that I am dividing by 306 because that is my expected value for spines. So after I divide, I get 0.64. That's going to be a different number that I have without spines because I'm going to take 196 and divide it by 102 because that is my expected value for my without spines phenotype. Once I do my division there, I should get 1.92. We are not done yet. Remember that you have to add those together. That's what that summation E stands for in our chi-square equation. So go ahead and add those values now. And you should get 2.56. At this point, you're done doing your calculation. However, we need to make sure that our math means something. So we need to now support or disprove our hypothesis and to justify our reasoning. So looking at our chi-square table, we are always going to use a p-value of 0.05. That is a 95% confidence level, and biology wants us to be at least 95% confident in our data. So that's our probability value or our p-value that we'll be using. And then I have the other smaller red arrow pointing to 1 degrees of freedom. 1 degrees of freedom is determined by looking at our possible outcomes minus 1. We have two possible outcomes, spines or without spines, two different phenotypes. So 2 minus 1 gives us 1 degree of freedom. And then when we look at 1 degree of freedom and a p-value of 0.5, we're going to see 3.84 is our critical value. So down at the very bottom of the slide, you can see how I wrote my conclusion and how I justified my answer. I have the chi-square value is 2.56, a p-value of 0.05, and 1 degree of freedom was used to determine the critical value of 3.84. because the chi-square value is less than the critical value, our hypothesis is supported. So that means that yes, we have data that supports that Mendelian inheritance and a single gene is responsible for pelvic spines in stickleback fish.